So welcome to the Brain Tumour Charities webinar series, and today we've been joined by David Jones and Mimi Bandopadhayay to speak about the paediatric low-grade gliomas and the translational and clinical implications of the molecular era. David is based at the DKFZ in Germany, where he's the head of the Paediatric Glioma Research Group, and he's also the lead of our Everest grant, which is the Paediatric Low-Grade Glioma Center. Mimi is based at Dana-Farber and Harvard. She's an attending pediatric neuro-oncologist at Dana-Farber and assistant professor of pediatrics at Harvard Medical School. We're going to start with David, and then Mimi will come second. So David, over to you. Thank you.
Okay, thanks very much for the introduction. Hope you can all see the screen okay there. Yeah, so thanks. Good morning, afternoon or evening, everybody, depending on where you're joining us from. And thanks for joining our webinar on pediatric low-grade glioma. In fact, I'm going to expand the scope a little bit and say low-grade glioma and low-grade glioneuronal tumours. I think they're really a family which are quite closely related, and a very important group of tumours in terms of overall childhood neuro-oncology. So if you take all paediatric low-grade gliomas and glioneuronal tumours together, then it's probably around a third, maybe even a little bit more, of all paediatric brain tumours occurring in this age group. And if we look back only 10 years ago, there was not so much that was actually known about both from the classification of these tumours and the molecular alterations causing these tumours. So in terms of the classification that was in use back then in 2007, there were relatively few entities that were described and no separation between paediatric and adult gliomas. And this is now starting to change, as I'll touch on a little bit later on. So in terms of molecular alterations, knowledge was pretty limited. There's been for a long time the established link between neurofibromatosis type 1, so germline mutations in NF1, with low-grade gliomas. And there'd also been some single reports of mutations such as the BRAF V600E or KRAS mutations. And we're just starting at that time to get some larger, slightly more focused studies looking at larger cohorts of these tumours and things like describing the BRAF fusion in pilocytic astrocytoma in 2008. But since then, there's really been a huge expansion in our knowledge, and the molecular era in many ways is overlapping with the next generation sequencing era. So there've really been many studies now focused on pediatric low-grade gliomas that have transformed our understanding both of the relationship in the different subtypes of the tumour and also the types of alteration that are underlying their biology. One of the most striking, I think, after focusing on BRAF for such a long time, was really this expanding role that we're now recognizing for FGFR1 in these tumours, either with point mutations at these two hotspot positions in the kinase domain, or with duplication of the kinase domain within the FGFR1 gene, or with a more canonical fusion, so where FGFR1 gets fused onto a part of another gene which is thought to be driving its dimerization. So multiple different mechanisms in which that gene can be altered, and it's starting to be more and more acknowledged that FGFR1 is playing an important role in multiple subtypes of pediatric low-grade tumours. But there were also a few other surprises waiting in coming out of the sequencing and the high throughput genomics era, one of which is that pediatric low-grade tumours are not just MAP kinase pathway diseases, or at least not associated purely with alterations in the canonical MAP kinase pathway members, So it was really a very striking finding when there were reports coming out of MIB and MIB-L1, the two transcription factors, fusions of those two genes also being involved in subtypes of low-grade glioma. So in angiocentric glioma and also in other pediatric diffuse gliomas. And really that was one of the first non-MAP kinase recurrent alterations identified, although as I think Mimi's is going to touch on in the second talk, even there they might still be having an effect through the MAP kinase pathway, just not in a typical direct way like some of the other tumor types. And we also started to see that these are not all single pathway diseases where you simply get one genetic alteration somewhere in the MAP kinase pathway here. So previously, and in pilocytic astrocytoma, it seems to be the case that these are all mutually exclusive to one another, and you only see one activating alteration as the main driver per tumor. But now we're getting examples such as rosette-forming glioneuronal tumor, which is starting to adjust our thinking there. And this is a very interesting example where, in addition to FGFR1 mutations, so pretty much all of the samples of this tumor type where we have sequencing data show one of those two hotspot kinase domain mutations in FGFR1. But additionally, about half of the cases also show a hotspot mutation in PI3 kinase, so in the PIK3CA gene. So not just the MAP kinase pathway, but also a direct genetic alteration in the PI3K pathway. And even some of them have this triple pattern where you get co-occurring mutations in FGFR1, PIK3CA, and NF1 all together. And some of those NF1 mutations are known to already be present in the germline. So very striking pattern, and certainly a different pattern of mutations compared to most of the other low-grade glial tumors where you would only see one of these events occurring in parallel. What we're also starting to see is that there's molecular evidence for novel entities emerging that were either completely unknown previously or had been hinted at based on histology and clinical features, but now also get a very clear molecular footing. And one example is diffuse leptomeningeal glioneuronal tumor. And this is another tumor which very frequently shows the KIA 1549 BRAF fusion, which is common in pilocytic astrocytoma. So this fusion, unlike what we may have been expecting, is not restricted purely to pilocytic astrocytoma. It can also occur in some other types of pediatric low-grade brain tumors. And here's an example where you see the duplication leading to the BRAF fusion. And what's also very, very frequently seen in this tumor type is this deletion of 1P together with the fusion. And that is almost characteristic of the molecular features of this tumor type. So what we're now trying to do is work towards an overarching classification based on these molecular features, or integrating the clinical, the histological, and the molecular features in terms of this integrated diagnosis. And the first hints that DNA methylation might be a useful tool for this came from the St. Jude's group in 2016, where they showed this very nice pattern in some FGFR1, BRAF, and MIB altered tumors, that genetic alteration, so you see the MIB here, BRAF in the middle, and the FGFR1 on the right-hand side, clearly overlaps with enrichment for certain types of histological diagnosis, but also very clearly shows distinct molecular profiles, as you can see on the right-hand side here. And when we expand this out now to encompass several hundred profiles from low-grade brain tumors, this still holds true that what we can see is these clearly distinct molecular subgroups based on their global DNA methylation patterns, which also overlap with an enrichment for particular molecular alterations and for a particular histological diagnosis. So this big corner here is primarily pilocytic astrocytoma and mostly with BRAF fusion. Then you see a small group corresponding to the rosette-forming glioneuronal tumours, which I already mentioned. There's a relatively recent subtype, myxoid glioneuronal tumour, which have a hotspot mutation in PDGFRA. And throughout this spectrum, you see this pattern recurring of a distinct global molecular phenotype with an enrichment for histology and an enrichment for a particular genetic alteration going with it. What we also start to see with plots like this are not just groups corresponding to some of those already described entities, but really things which point us to completely unknown tumour classes. There's one such group in the middle here, which didn't show any clear overlap with a previously known entity. And when we look in more detail at the tumours within this group, what we see in this group, and we now have more than 60 cases collected within this group, two very striking features. Firstly, that they have an extremely broad morphological spectrum. So this might be the main reason why they haven't been identified as a unique tumour group yet, because they don't show very clear distinctive pattern under the microscope. But they do have this distinctive methylation profile. And all of the cases that we've sequenced to date show a fusion of this gene here called PATS1, either with MN1 as an upstream partner or with the Ewing sarcoma gene EWSR1 as the partner. They're a very interesting new tumour group, pretty even split in terms of the gender balance, mostly found in the supratentorial regions, but also some examples throughout the CNS. And in terms of survival here, although the case numbers admittedly are still very low, what you can see is that they do quite often progress, but the overall survival is not quite as bad as what you'd expect of a typical high-grade tumour, and might be suggesting that it belongs within this low-grade family. So this is interesting because many of these were histologically diagnosed as very malignant high-grade tumours, so glioblastoma or PNET, and they don't seem to be behaving as poorly as you would expect based on that in terms of the outcome data that we have so far. Many of these advances are now being introduced into the upcoming modifications to the WHO classification. And this is now well underway, the discussions for the new version of the WHO. And what's very nice to see is that there is now a completely distinct chapter included in the classification called Paediatric type diffuse low-grade gliomas, as well as also Paediatric type high-grade gliomas. So really now finally seeing an acknowledgement that our paediatric patients are not just small adults in terms of the tumours that they're developing, but that we really need to treat these as completely distinct family of tumours. So here are some of the entities in that paediatric diffuse low-grade glioma group. So diffuse astrocytoma, MIB or MIB-L1 altered, angiocentric glioma, mostly QKI MIB fused, very nicely confirmed in the mechanism of that, shown by Mimi a few years ago. Polymorphous low-grade neuroepithelial tumour of the young, which often display FGFR2 or 3, so other FGFR family fusions. And then this broader category of diffuse low-grade glioma, MAPK pathway altered. And also outside of that pediatric specific chapter, there are still some pediatric and adult mixed chapters, but there's also been a number of new additions that will be made. For example, within the circumscribed astrocytic tumours, is now high-grade astrocytoma with pyloid features, which is essentially what was previously known as anaplastic pilocytic astrocytoma. Another MN1 altered tumour, so this is not the same as the MN1 PATS1 fusion that I just told you about, but another group of MN1 altered tumours, and then also some new additions within the mixed glioneuronal tumours. And what's nice is that for many of these groups now, we can also add on in terms of molecular annotations that we know in many of these cases what is also the underlying genetic event. So pilocytic astrocytoma, various MAP kinase alterations. The higher grade tumours have those MAPK plus CDKN2AB deletion and HUX loss. Pleomorphic astrocytoma with the V600E and the CDKN2A and B loss, and so on. So you really get now this clearer match between a global molecular pattern, a histological pattern, or sometimes a histological spectrum, and distinct molecular and genetic alterations that are clearly enriched in some of those groups. And that's starting to be used now. This combination of different features is starting to be used for risk stratification of low-grade tumours. So I really wanted to pick up on this fantastic paper which came out earlier this year from the groups at the Sick Kids, so led by Uri Taburi and Cynthia Hawkins, and also a big contribution of David Ellison and St Jude's, with molecular and clinical data on more than 1,000 pediatric low-grade gliomas. So really, really impressive study. And they suggested this risk model now for determining risk both of overall and progression-free survival split into these different groups. So within the low risk category would be the BRAF fusion, NF1 alterations, MIB and MIB-L1, FGFR1, either the fusion or the duplication, and FGFR2 fusions. An intermediate risk would be IDH, V600E, FGFR1 mutation. So that's interesting now that the particular way in which FGFR1 is altered might also be modifying the risk here, and MET mutations. And then high risk, the very rare histologically low-grade tumours that do occasionally show the histone 3, K27M mutation, and those tumours with the V600E together with the CDKN2AB deletion. And you can see those split, particularly the PFS, but also the OS nicely. We've now looked at that model and some other features together with some of our own data, and you can sort of split this also in other ways and get similar risk stratifications. So if you take, for example, the K27M mutation or any CDKN2AB deletion as high risk markers, intermediate with IDH1 mutation, and all tumours regardless of genetic alterations which can't be completely resected surgically, and in the low risk, all other low-grade glioma groups. And you also get this split both in terms of PFS and OS. So some of these models now really need to be prospectively checked and controlled in some of the upcoming trials. So in terms of diagnosis, there's a very wide spectrum of alterations in these tumours, many of which are potentially targetable or druggable. But in order for those to have clinical value, you have to be able to detect them. So we now need to be using broader platforms than just single gene testing, particularly for those cases which have an unusual or unexpected histology. It's also important to be aware of those associations between unusual types of molecular alterations, FGFR2/3, chromosome 1p loss, which might be supportive of an alternative diagnosis in comparison with what was originally expected. So there's a minimum now. It's necessary to test for BRAF mutation and fusion, FGFR1 mutation, fusion and rearrangement, MIB status, and also CDKN2AB and H3 in order to exclude the high-risk tumours, even with low-grade histology, and arguably more high-throughput techniques such as copy number profile, DNA methylation, and wider sequencing approaches are extremely important to add on in terms of really getting to optimal clinical decision-making. And many of these are now going to be required in order to stay compatible with the new WHO classification. For clinical trial planning, that means you need very clear structures in place of how that profiling is going to be done. So within the European-wide LGG study is now a bioclinical data bank, which is already open in Germany and soon opening in several other countries to centralize the performance of that molecular diagnostic testing and allow for things like prospective testing of the risk models, and being able to match that intensive molecular data with the outcomes from the clinical trials will be an extremely valuable resource for optimizing some of those biomarkers going forwards. And not just in phase 3, but also in terms of early phase, some of the new insights that we're seeing in these tumours are also opening up options for new phase 1-2 studies in paediatric low-grade glioma, either directly targeting some of those alterations, for example, the FGFR1, or maybe some of the PI3 kinase mutations, but also in terms of phenotypes or mechanistic features. So there's now interest in combining the MEK inhibitors with a class of compounds called senolytics. And the hope is that through that combination, you target not just the cycling MAP kinase activated tumor cells, but also the senescent sort of hibernating treatment resistant cells to more rapidly reduce the tumor burden. So to sum up, this is really an extremely diverse tumor group, and much more diverse, much more that we've learned in the past few years than we could have imagined even a decade ago. Many of these novel classes now have potentially direct therapeutic relevance in terms of being targetable, but you need to be able to identify those alterations. So you need ways of providing a detailed molecular diagnosis. And the future clinical studies will now focus both on investigating the effect of novel treatments, but also confirming through the larger phase 3 studies some of the prognostic biomarkers that have been suggested. So with that, I'd like to thank my group, but also many, many other colleagues and collaborators around the world who have made this such a fascinating field to work in over the past few years. And I'm going to end while I introduce Mimi with a bit of shameless advertising. So although not taking part in the real London Marathon this year because it's not happening, I don't count as an elite athlete, unfortunately, I will be running 26 miles for the virtual London Marathon on behalf of the Brain Tumour charity. So if anybody can spare a pound, a euro, or a dollar there, I very much appreciate it. So I'll leave that up while introducing friend first of all, and colleague as a close second, Mimi Bandopadhayay from Boston, who's done incredible work in this field over the past several years, and she's going to give us a taster of some of those new insights from her lab in the next 20 minutes or so. So over to you, Mimi.
Thanks, David. So we are having a step competition right now at Dana-Farber as to which lab can walk the most. I'm about to recruit you to my lab because if you're doing the marathon, I think you're a totally Bando lab for that. So thank you for that really thorough and wonderful introduction to the genomics of low-grade gliomas. So I get my slides up. Because of really these discoveries that have been made over the last decade, pediatric low-grade gliomas have become the poster child for precision medicine approaches in pediatric oncology, and that is incredibly exciting. Before I launch into the science, I have a quick disclaimer slide of conflicts of interest. I received grant funding from Novartis and a consultant for QED. So as David sort of really mentioned, we've learnt more about low-grade gliomas over the last decade than we have about many other cancers. And it is really exciting. And it's really exciting to think about all of these targeted inhibitors. But it is also really important to think about the biology of low-grade gliomas, and in particular, the clinical course that we see in these children. And they are very different to adult gliomas. So David just showed you how they're different genomically, but clinically they're very different as well. So an adult patient who is diagnosed with an adult low-grade glioma, most commonly an IDH mutant, undergoes years of stability, but then the tumors transform and the majority of patients die of their disease. We fortunately don't see that same course in kids. So low-grade gliomas, children are most likely to be long-term survivors of their disease. And when we look at their outcomes many years out, so I'm showing you here a Kaplan-Meier curve with many, many years of follow-up, looking at what happens to adult survivors of low-grade gliomas, we don't see the same levels of malignant transformation. Adult survivors of this disease are going to die of things that adults die of. They don't tend to die of low-grade glioma. So that's the good news. And then when you look at actually what is the course of a child who's diagnosed with low-grade glioma, what is the likelihood that they need interventions and more than one intervention? And this was a really informative paper that was published by the SIOP group last year, trying to model the natural history of these gliomas. And I'm just going to point out this as an example here. So in this plot, they were looking at all of the children that had been diagnosed with a low-grade glioma that had needed a surgical intervention and then had needed some sort of treatment, chemotherapy. And they were modeling the prediction that that child would need something else after that treatment. And the good news is that 3/4 of the kids were not likely to go on and need a second treatment. So about 1/4 of the kids, if you've been treated, for example, with vincristine carbo, need something else, but the majority don't. And I think that is a really important message for us to remember as we start thinking about introducing these targeted inhibitors into the upfront treatment for kids with low-grade gliomas. That's not to say that low-grade gliomas don't cause devastation in other ways. You can have a tumor growing and not need, for example, treatment, or you can have a tumor growing and need one course of treatment that still leaves you absolutely devastated. The tumors cause more too often than we want to see effects in the kids, their growth, their vision, their cognition, endocrinopathies. The chemotherapies that we use give them side effects, and we see long-term side effects. So it's a paradoxical tumor where we tell the families, the good news is your child is likely to survive, but it's a tumor where, you know, more than anything, survival isn't the only outcome measure. We want to get these kids into their early 20s, into their adult years with minimal side effects, no side effects, right? We want them to be able to do what they can do. As David said, these are a heterogeneous disease. Genetically, they're absolutely heterogeneous, but even in terms of what the pathologists see under the microscope, before we knew about these drivers, the pathologists could tell us often the different subtype by looking at the morphological features, but we frequently and still have children where the tumors are low grade but don't have morphological features that allow the subtyping into one of the histological subtypes. And then, as David said, they have a huge diversity of genetic drivers, but they are unified by the fact that these alterations activate a common signaling pathway or pathways. And so they all converge on the MAPK and mTOR signaling pathways. And in many ways, we were lucky, or that was good news for us, in that these same pathways are activated in other cancers, particularly melanoma, and drugs had been and continue to be developed for melanomas that have proved to be very exciting for children with low-grade gliomas. And so there are an increasing number of medications and targeted inhibitors that are being considered or in use for these kids, but they tend to, at the moment, focus in on ways to inhibit the MAPK signaling or the mTOR signaling pathways. So when you see that, if you think about a child has come to your clinic with a BRAF mutated tumor, it seems really exciting, right? There's all of these BRAF inhibitors that people can use. But it's really, really important to understand biology, and particularly when you start to use drugs that can inhibit pathways and can result in activation of other pathways or affect normal development. And low-grade gliomas taught us this as well, in that if you don't understand the biology before starting to use these drugs, you can actually do exactly the opposite to what you are hoping to do. And so this is an MRI scan from a child that was treated on a clinical trial a few years ago, which was evaluating a kinase inhibitor called sorafenib for children with low-grade gliomas. And the trial was stopped early because of cases like this, where after the drug was initiated, so here's the child at the start of the study, within only 3 months, there had been significantly increased growth of the tumor. And that was because this drug and other drugs that inhibit BRAF can actually cause paradoxical activation of the signaling pathway in the presence of a BRAF rearrangement, which is the most common type of alteration that we see in low grade. So it's nowhere near sufficient to be able to say this gene is mutated, therefore we can use this drug. We really need to understand the biology, and in clinic, when we think about using these inhibitors, we really need to know what the mutation is for the child sitting in front of us in clinic and really tailor the use of the inhibitors to that child's mutation. Having said that, the targeted inhibitors, when used appropriately, have been really showing early clinical promise for kids with low-grade gliomas. And there are BRAF inhibitors in the clinic, there are MEK inhibitors in the clinic, there are mTOR inhibitors and other drugs. This waterfall plot that I'm showing you is actually one of the MEK inhibitor studies. This is the selumetinib study. When you look at these waterfall plots, 0 is the baseline of the tumor on enrollment. When you see it going up, that's growth of the tumor on the drug, and when you see it going down, that's a response. And so you can see this is really exciting. In this trial of about 25 kids, the majority of children had either stabilization or good responses to this MEK inhibitor. This plot is actually color-coded with the mutations. Blue is the rearrangement, and the pinkish color is the mutation. You can see kids with both alterations are responding. However, kids with both alterations also didn't respond, and we at the moment do not understand why some kids respond to the drugs and why other kids don't and have a good biomarker of response. So as these drugs continue to go into clinic and as we get increasingly excited about them, it's really important for us to keep thinking about what are the unanswered questions and what are the challenges. And here I've listed just a few. David alluded to the fact that the discovery of the MIB alterations really was the first time where we found mutations in low-grade gliomas that did not converge on canonical MAPK or mTOR signaling. And he just showed you another example of the PATS1 alterations. It is still an unknown question as whether we would see similar responses to the inhibitors with kids with these mutations. Having said that, we and others, including David and his team, have evidence that expression of the MIB alterations is sufficient to activate ERK signaling, and to a lesser extent mTOR signaling, and we're trying to actively really understand whether we should be using these inhibitors. As David also said, the majority of kids with low-grade gliomas only have one alteration, which is exciting because that's probably a big part of the reason for why we're seeing these responses. But what happens if the tumors have two mutations like we are increasingly seeing with the FGFR altered? If you inhibit one of these alterations with, for example, an FGFR inhibitor, is there a risk of getting paradoxical activation of some other signaling pathway? So we're trying to address that. But for the rest of this talk, I'm going to actually focus a little bit on some issues that I think we need to, as a community, really try to understand urgently, because we are using these drugs in clinic right now, and there are some questions that really need to be answered relating to resistance and biomarkers, but also trying to understand how the use of these drugs may actually interplay with the normal history of these tumors and also effects on development. So the inhibitors, if you actually think about the timeline, the rate at which we've learned about low-grade gliomas in the last decade is absolutely exciting and inspirational. Before 2008, we didn't know what the genetic drivers of these tumors were. We had an idea that they were part of the mTOR MAPK signaling because of the kids with NF1 or TS tuberous sclerosis. But those mutations were only discovered just over a decade ago. And in that time, we've gone from discovering the alterations to having clinical tests to drugs being used in clinic to the early phase clinical studies being completed. And I think that is really quite remarkable. But we are starting to learn that it's not going to be as simple as can we just use these inhibitors and get good responses. There is early evidence, and these findings need to be confirmed and are being confirmed in collaborations across doctors from all countries and all continents, but there seems to be an observation that when we treat these kids with these inhibitors, we get the promising responses that we see. So here is a child who has a BRAF mutated tumor growing in his temporal lobe. He was started on a BRAF inhibitor actually and had a really good response like we're seeing. But after we stopped treatment, within only a month, not only had this spot grown back, it had grown back to larger than it was before we started. And so we are starting to anecdotally see this evidence of what we call rebound growth, which is when we're stopping the drugs, whether it's a BRAF inhibitor, a MEK inhibitor, a targeted inhibitor, some of the tumors in some of these kids tend to be growing back. If we re-challenge them, we can see responses again. And that in many ways sort of starts to suggest that maybe the tumors are becoming addicted to these drugs, and when we're releasing them, we're releasing the brake. And so what our challenge really is to understand why is this happening, and then try to find ways in which we can do something here to stop that rebound from happening when we stop the drug. So to try and understand this, we need to understand the biology of the tumors a little bit more, and then with more granular detail. And so we and others have been applying single-cell RNA sequencing to try and understand the cell programs of these tumors and the cell states that make them up. And this is an example of 6 pilocytic astrocytomas. These all have the BRAF rearrangement of fusion and profiled with single-cell RNA sequencing. On the x-axis, we're looking at the differentiation state of these cells, so astrocytic-like or oligodendrocytic-like. And on the y-axis, we're showing the level of MAPK signaling. And what we find with the low-grade gliomas is that they tend to have already committed towards a astrocytic differentiation. So they're more astrocytic-like than oligodendrocytic-like. There is a population of cells that are a little bit more stem-like, not as stem-like as what we see in higher grade glioma. So they're already committed to part of this differentiation. But there's only a subset of these cells that are actually dividing, as shown in red. So these are cells that are cycling. And what we're trying to understand now is if we take these tumors and put drug on it, do we shift these cells down into this either more differentiated state, or I'll show you another state that they might go into where they're not dividing, and then is there plasticity? So when you release the drug, are they then able to just go back up and start dividing again? So that's something that we're looking at. The other thing that is really, I think, absolutely important for us to keep thinking about is if we don't treat any child with a low-grade glioma with these inhibitors, they will be expected to get really bad side effects from the tumor growing, but they don't tend to undergo that transformation that we see in adults. And why is that, right? Why is it that these low grades don't acquire P53 mutations or other alterations and become a higher grade? And is it actually because of the fact that these are tumors that are growing in the context of a developing brain, and does the signaling of the normal brain actually signal these tumor cells to actually stop dividing when kids become young adults? And so we're trying to look at this a little bit more again with our single cell RNA sequencing. I showed you this plot before with the red dots, which are the cells that were cycling. But when we actually look at all of the cellular programs that are most correlated with MAPK signaling, we actually find that the #1 pathway that is most correlated is actually senescence. So this is a normal brake mechanism that normal cells have when you acquire signals that make cells divide faster on the path to becoming a cancer. As a protective mechanism, cells often become senescent. They, as David said earlier, go to sleep and don't divide. We call that oncogene-induced senescence. And because the majority of low-grade gliomas don't have other mutations that other cancers have to escape this, for example, P53 mutations, we think that a lot of the cancer cells in the low-grade gliomas are actually subject to this oncogene-induced senescence. And we see similar things in our models of low-grade glioma. So here are 2 models of neural stem cells that have been transduced to express the 2 BRAF alterations. These cells are not being passaged in any other growth factor because they're dependent on these alterations. And we see similar levels of cycling and senescent cells. And that has led us to this model that we're trying to prove or disprove in the lab at the moment, which is that in low-grade gliomas, and potentially other cancers as well, you know, for example, the nevi that become melanoma. We think that there is a sweet spot of MAPK or mTOR signaling that is needed for a cell to grow faster than normal and increase its proliferation to become a tumor. But if it goes too high, you induce oncogene-induced senescence, and that actually slows down the growth of the tumor. We see some evidence of this in the tumors. So when we go back and look at the RNA sequencing data, we find that these tumors are starting to turn on other genes that actually dampen expression of MAPK and mTOR signaling. So here are two genes, TSC1 and TSC2, negative regulators of mTOR signaling. And when we look at normal brain versus low-grade gliomas, these tumors tend to have higher expressions of these negative feedback loops, suggesting that they might be potentially being expressed to try and really bring the cells back into that sweet spot. We have been interested in this and sort of hypothesized that if the cells are turning on negative regulators to keep them in the sweet spot, maybe if you delete them, then the cells won't like that. And so we've been using genome-scaled CRISPR screens to try and identify vulnerabilities that are introduced with expression of these oncogenes. So here what you do is you take a model of a low-grade glioma, you introduce them with CRISPR libraries that can go in and ablate every single gene in the genome in individual cells, and then you can compare these dependencies. So genes that are required for proliferation, those cells are depleted. So here are results of two CRISPR screens of a normal cell line and a low-grade glioma relevant cell line. And a negative result here is a gene that is a dependency. So what we are interested in are genes that are dependencies in the low-grade line, but not in the normal line. And what we actually found were that some of the top hits of dependencies in the low-grade lines, but not in the normal cells, were actually these negative regulators of mTOR signaling. Again, so, you know, it doesn't prove anything yet. We're trying to work on this, but it adds some weight to our hypothesis that the cells might be doing everything they can to keep their level of expression in this sweet spot. But low-grade gliomas don't grow in the tissue culture dish, right? They grow in the brain in the context of a developing brain. And so to really understand that, we need to understand what's going on in the normal brain. So we paired up with Claudia Kleinman and Nada Jabato at McGill, who have recently analyzed normal developing mouse brains with single-cell RNA sequencing. And what their data suggested, and as what has been shown before, is that the level of MAPK signaling is the highest in glial cells, so cells from which that are, you know, from which gliomas tend to arise. And that's not surprising because these pathways have been shown in the past very nicely to be important in determining the glial fate. What we also found when we looked at that data is that the absolute level of MAPK signaling also increases with chronological age. So the level of MAPK signaling in the entire brain was the highest in the adult mice compared to embryonic or young postnatal mice. And so if we take that back to our model, we think that the low-grade glioma cells increase the level of signaling to maintain proliferation, but if it goes too high, you become senescent. Well, what happens if your brain is developing? If the background level of MAPK signaling in the developing brain is increasing, then maybe that is sufficient to induce this senescence, which is causing quiescence and then stability. And we're trying to urgently prove this or disprove this in the lab, because the underlying question that I have is, if that's the mechanism through which these tumors are stabilizing and potentially ending up with a more compact chromatin landscape that is not as conducive to acquiring second mutations, how will the use of these inhibitors actually affect this? And is there a risk that we might actually interfere with that normal quiescence and actually change that natural history? I'm hoping this hypothesis is wrong, but it's something that we, along with David and others, are trying to really actively address, because as David said, we are now starting the clinical trials that are testing these inhibitors in the upfront setting. And then the other question that's always there in pediatrics, particularly with this pathway being so important in development, is how will these inhibitors affect normal development? And then it's not all about just the tumor cells themselves. These cells are growing, as I said, in the context of a developing brain. And we've been trying to understand what are the other populations of cells that make up these tumours. Again, a really exciting collaboration with multiple people across multiple continents. And what we are starting to find and starting to try and understand is that in addition to the tumour cells, these cells have a large infiltrate of immune cells, inflammatory cells, and particularly as we're profiling more and more tumours, large populations of microglia. And their role in interacting with the tumour cells and the potential for therapeutic targets still remains to be explored. So in summary, we have shown that low-grade gliomas are a heterogeneous disease. They have a very distinct clinical course, but they are generally characterized by stable genomes that activate MAPK and mTOR signaling. They do exhibit transcriptional heterogeneity, and we're trying to really understand how this interplays with inhibitors and how to optimize the use of inhibitors. And then in the last sort of few seconds, I want to thank everyone in my lab who is working on this, but also a very large team of collaborators. One of the absolute joys in working in pediatric oncology in this field is the ability to work with people from all across the world and the absolute collaborative spirit that there is. David is, I will also call you friend first and collaborator second. It's absolute joy to work with, and it's really wonderful to have a team of people that are on this mission to try and find better treatments as quickly as we can. So with that, David and I are going to take some questions.
Well, thank you both for some very interesting and thought-provoking questions as presentations, as we can see by the number of questions that have rolled in through that period. First of all, though, I want to take Chairman's privilege and ask the first question, and that is, given the context of the NHS in the UK and our ability to or to not get some of these tests because they are not reimbursed currently. Did I understand you correctly, David, that the WHO, the World Health Organization, are going to look at including things like methylation in their classification and the use of, despite the fact it's non-universal availability, and that being a reason they've given in the past for not doing so?
Yeah, so as I understand it, there will be a system now of, for each entity that's defined there, essential and recommended diagnostic criteria. So it won't just be a yes or no. There'll be some criteria that the tumour will have to meet to be able to fit into that group, but also additional levels on top of that, which provide strong supporting evidence, but they're not essential criteria. So the goal of the essential would be that that's as broadly applicable and as globally applicable as possible. But so not just methylations, other single gene tests, things like IDH, 1p19q test status, very others which have been sort of on the periphery for a while, will now be in this sort of highly recommended, or for some of them. I think for very few of the groups, they will really be essential as well, for certain molecular tests. But the majority, those molecular tests will be in that second level of strongly supporting, but you can make the diagnosis without being able to access those tests.
I should say, if you do have any more questions for David or Mimi, please add them to the chat. We've got about 10 minutes of time, which we'll use to get through as many of those as we can. So maybe to take it the step further then, and this is a two-part question. First part, I think we'll throw towards you, David, and that is, what do you do if the histology and methylation classes are controversial? For example, a tumour with high-grade features such as necrosis, mitotic activity and vascular proliferation, but a methylation class of RGNT. What's the advice on the diagnosis?
So with the caveat that I am not a clinician and not certified to make any kind of official diagnosis, my personal view is that the methylation analysis is an extremely powerful tool, but I would never use purely that as the one thing that overrides everything that you're seeing from all other pathological studies you're doing. So if all you have is on one side, everything says it's a glioblastoma, and then the methylation says something else, I don't think that would be enough to overturn it. But if you also have methylation says low-grade tumor, and you have BRAF fusion, no CDKN2A deletion, no P53 mutation, and the child's had a history of seizures for 3 years, and etc., etc. So it's a bigger picture where methylation can play a part in adjusting a diagnosis if those things fit together. And we definitely have seen examples where very classical histologically high-grade tumors can have biologically and clinically low-grade behavior. So they do exist, but you would need a body of evidence to support that change to a low-grade diagnosis, not just purely the methylation without anything else supporting it.
And as the clinician, I'm just going to totally concur with David. And we would, I don't think that any one genetic test by itself is sufficient. The methylation adds support to it, but as David said, you need the mutations and the copy number profiles as well, because some of them will have very distinct high-grade signatures that will help you with that classification or diagnosis.
I think it's particularly important in infants as well. So that's where these examples come up most often, is where you see histology, terrible high-grade GBM, but then clinically they do much better. And we're starting to learn that a big chunk of those infant tumors might just be completely different to either a high grade or the typical low grades, and are essentially their own thing. And that's where you need to be really careful of trying to collect as much evidence as possible.
Thank you. Going into the single cell work you've done, Mimi, a question here about the ability of cells to travel from one of those, the cycling to the resting to the quiescent or senescent compartments. How do we know that dividing cells on the day of the single cell RNA assay would not become non-dividing if tumours were left intact? You know that?
So actually, I think that's what happens, right? That's my hypothesis for why I think these tumours stop growing when you become a young adult, and why they don't transform. I think as the activity, as the brain, the normal brain is developing and the levels of MAPK signaling go up in the developing brain, that induces senescence, and that stops them from dividing. So that's exactly what I think happens. What I don't know is how these drugs will interplay with that.
Okay, excellent. One for you, David, because I know it's part of the Everest grant work, is around cellular models and lines. Are they available for these pediatric low-grade gliomas, and what types are available?
There are a couple. They're sadly rare as hen's teeth, so it has been extremely difficult in the past to get these to grow in culture because they're low-grade tumours and they senesce or quiesce and don't like growing outside of their microenvironment. There's now, I think, a nice NF1 model from Charles Eberhardt's lab. There's a BRAF fusion model from the group of Olaf Witt as well, and there's one or two others that are coming up. So there's huge efforts ongoing around the world to try to expand that range. What you can do as a sort of halfway house is some of the approaches that Mimi's been taking is to exogenely express the genes in another system. So put the driver genes from the low grades into a neuronal stem cell, which is a little bit more artificial, but still allows you to investigate the mechanism. I'm probably going to get shot for saying this, but we do think that we might have now got one additional BRAF fused PA line growing recently in Heidelberg. So we're getting there, but it's hard work.
Mimi, are there any mutations that drive different types of LGG which provide opportunities for immunological targeting via vaccines or other routes?
Yeah, that's a good question. I mean, we've thought about this before. For example, could you design a vaccine, for example, to a fusion protein? I think it's hard. A lot of these fusions have different breakpoints in different patients, so it's hard to have one consistent approach. What we're trying to understand, and I know a lot of people are, is we actually don't understand the responses or the interactions between tumour cells and immune cells here in these tumours at the moment, and potential for that. So I think some of the, PD-1, PD-L1 inhibitors that have been really exciting for some of the other solid tumours, we don't tend to see as much expression in these tumours, but there might be other ways that these tumour cells are evading the immune system that might have therapeutic potential later. So as we're increasing the number of tumours that we profile, we are keeping all the immune cells in the single cell data set so we can try and understand that.
And I don't know who wants to take this one, but what's the role of vascularization in low-grade glioma therapy? I will pick if someone doesn't step in.
We're both looking at each other.
Good question. I think the short answer is we don't really know. There have been some studies, and Avastin particularly has been used in some contexts. I think probably Mimi can speak more about that than me. We know that they do show microvascular proliferation, and in terms of contrast on MRI and other features, we know that it's abnormal and that there are alterations in the tumours. But I think therapeutically, it's not something that's been explored to the fullest extent yet. Avastin in some contexts helps a little bit, but it hasn't been a blockbuster.
Yeah, I think Avastin is probably the closest that we've come to trying to use it therapeutically. Yeah, it's not something that we've really focused in on a lot.
Okay, I think we've got time maybe for one, possibly two if we rush more questions. Do you think the prevalence of the KIA 1549 BRAF fusions in paediatric low-grade glioma is related to the functioning of these genes and perhaps chromatin features in the developing brain that predisposes to the fusion mechanism, or are we only seeing what is selected for in a tumour? David?
Both. So I think there is probably some good evidence that chromatin architecture is really playing a major role in that occurring. And if you look at, so although you can get different breakpoints at the genomic level in the KIA 1549 BRAF fusion, there are some exons which are much, much more commonly associated with each other than others. And those introns around those exons are marked with certain chromatin marks during different stages of evolution. And you see differences in the frequency in the exact type of fusion breakpoints in different regions of the brain, which also suggests that the precise chromatin architecture in different brain regions might also be affecting which tumor, which fusion you're more likely to develop. But I think always there's some aspect of the tumor also has to firstly survive that alteration occurring and then have other features which allow it to proliferate once it does occur. So I think it's a mixture of both of those.
Yeah, I agree. I think as we're looking at rearrangements in general across the board, definitely in low-grade in pediatric brain tumours, these tumours find ways to hijack normal developmental signaling pathways. And a lot of these fusions, or many of these fusions, are actually juxtapositioning or juxtaposing a gene that is normally expressed in the normal brain and essential for development with a gene that's not, to turn it on, right? So this enhancer hijacking to turn on a gene when it shouldn't be on. We haven't really worked it out, that mechanism for the KIA 1549 BRAF, but there are definitely differences in the expression patterns of these genes during development that might be intervening here. But for things like MIB and some of the other alterations, it's very, very obvious that that's what's going
on. So I think, we are over time and obviously people need to get off to other things, but a very quick one for you, Mimi. Tumour treating fields, we've seen them have an effect in adult GBM. Is there anything that they could do here, do we think? Is it something people are looking at?
I saw Dr. Monge was logged in. I don't know if she's still here. So it's not something that we have personally looked at, but there is really interesting data coming out from Dr. Monge and her team about the interactions and signaling between normal cells and tumour cells. And so I can't see her name, so I think she might have logged off. I think potentially we haven't looked at that, but that could be not necessarily for the current devices that are out at the moment. I think more work needs to be done, but trying to really understand some of the paracrine signaling between tumour cells and normal cells outside of what we are would be really interesting.
Well, I think we're out of time, so I'd like to thank you both again for the talks and for answering the questions. Maybe we'll be able to send you the ones that we haven't been able to get to, and we'll be able to put something on the website with the recording when it goes up. So all that's left to say is thank you to everyone who's logged in today. The next webinar is on the role of extrachromosomal DNA in cancers. That will be on Wednesday, the 7th of October. I believe registration for that will open shortly, so please come along to hear from Rolver Hack around that topic. And we always look for feedback around these webinars. It's something that we started through lockdown. We've found them very useful for ourselves, but we also hope that the community is finding them useful as well. So please do feedback to us. Please let us know if there's any other topics you would want us to cover and anything like that. So without any further ado, thank you to Mimi and to David for your input today. Very informative as ever, and thank you to everyone, and we will see you at the next one. Thank you.
Thanks everyone.
Thanks. Bye.
Bye.